My delegate ID is 612. My name is KV Prabhu. I am from Thoothukudi Government Medical College. My topic is on neonatal septicemia at in hospitals. Introduction. Neonatal septicemia remains the single most important cause of neonatal mortality, and it holds the first rank in that neonatal mortality. In India, neonatal septicemia accounts to 10.97 to 27 per thousand live births, holding the first rank in neonatal mortality. Research questions. What is the incidence of neonatal septicemia? What are its antibiotic sensitivity and common organisms prevalent? Aim of the study is to identify the common bacterial pathogens associated with neonatal septicemia in a neonatal unit and to study the susceptibility patterns and common organisms present. Materials and methods are total number of samples is 45 samples. My study period is from June 2008 to November 2008. The study area is pediatric department of Thoothukudi Government Medical College. And the sample, the 2 ml of blood was collected from the infective neonates under aseptic precautions. The blood was inoculated into culture media and with 20 ml of brain heart infusion. Subcultures were made on 5% sheep blood agar and McConkey agar plates, and isolates were identified by using standard biochemical tests. Antibiotic susceptibility tests were done using a modified Kirby-Boyer disk diffusion method. The results are out of the total 45 samples, 34 samples showed growth and 11 samples showed no growth. The predominant samples are staph aureus, which showed 12 out of the 34 growth samples. Next comes the Pseudomonas and Klebsiella, which accounts for 6 samples. Then Citrobacter and Acinetobacter for 5 and 3 samples respectively. E. coli and cons came up 2 and 1 samples. So this is the picture of the Staphylococcus aureus, and it's showing the culture, showing the circular white shiny opaque colonies, and it's the tube coagulase test confirming the presence of Staph aureus, and there's a culture showing the Pseudomonas green pigment on nutrient agar, and Klebsiella showing pure leukoid colonies on McConkey agar. Antibiotic sensitivity research. Staph aureus. For Staph aureus, ciprofloxacin and erythromycin are mostly sensitive and resistant to cotrimoxazole, amoxicillin and cephalexin. Pseudomonas, most isolates were sensitive to ciprofloxacin, amikacin and cephaloxim and resistant to amoxicillin. My conclusion is, study identified with common bacterial pathogens and their antibiotic sensitivity patterns in our pediatric neonatal unit, and the results showed most isolates are sensitive to ciprofloxacin and amikacin, and Staph aureus was the predominant isolate in our study. This study emphasized the importance of understanding the local epidemiology of neonatal septicemia in our area and to formulate a new antibiotic policy. Since for neonatal septicemia in our hospital, ampicillin and cephaloxim is being given, but since most of the strains are resistant to amoxicillin, the drug won't be effective and it may even become fatal for the neonate. So my future perspective is to study the ESBL resistant species among the gram-negative isolates. Neonatal septicemia is a life-threatening situation and should be handled with care. So proper antibiotic sensitivity tests should be taken, and with those results only we should apply. Thank you very much.